Dear learner, welcome you all in the course of Immunology BZYET 141. It is a elective courses for BSc General Program. Dear learner, my name is Dr. Siaram. I am an Associate Professor, Discipline of Life Science, School of Sciences, IGNOU. And dear viewer, today I want to discuss a very important topic that is known as the working of the immune systems third. Basically, it is a very important chapter of your courses. And my what is the expected learning outcome of this session? I want to discuss one by one. Define after completion of this session, you will be able to define the T cell lymphocyte and T cell mediated cellular immunity. After finishing of this course, you will illustrate the structure of the CD4 positive T cell and CD4 positive CD8 positive T cell. After finishing of this session, you will be able to differentiate between the cytotoxic T cells and helper T cell. These two T cells which are very important, and they are involved in the T cell mediated immunity. And after finishing of this session, you will be able to describe the T cell positive selection and negative selections. So what is the positive selection of the T cell and what is the negative selection, we will discuss in detail. And after finishing of this session, you will be able to discuss the process of the T cell differentiation, maturation and activations. And finally, we will discuss the memory cell development, how the memory cell develop after infections. So these are the expected learning outcome of today's session. Dear learners, my session is divided into two parts, part first and part second. In part first, I will discuss in detail what is the T cells, how T cell involve in the cell mediated immunity, T cell receptors, and how classified the T cells and antigen presenting cell, which are very, very important, they can involve in this T cell mediated immunity. In part second, we will discuss the T cell activation, T cell maturation and T cell differentiation, and finally we will discuss in detail the memory cell development, how the memory cell develop after infection. So I want to discuss one by one in part first. In part first, I want to discuss the T lymphocytes and T cell mediated cellular immunity. First of all, Jacques Miller and his PhD student Dr. Graham Mitchell discovered the T cell in 1963. It means the first time T cell was discovered in 1963 by the Jacques Miller and his PhD student Dr. Graham Mitchell. And T cell mature in the thymus. I have already discussed in my previous lecture. It is mature in the thymus and participated into the cell-mediated immunity. The number of the cell-mediated immunity are there. In this case, the T cell involves and antigen-presenting cell are involved. In T cell, prevent infection or destroy the intracellular antigens like viral and bacterial infected cells are there. It will involve into the cell mediated immunities, and T cell, antigen presenting cell, and macrophages, NK cell and antibody dependent killer cells that work in coordinating manners and provided to the cell mediated immunity. So basically cell mediated immunity, it is it work under the coordination manner. So number of the cells are there, like B cell, like antigen presenting cell, like macrophages, like NK cells. They work in coordination manner, or ultimately they are able to provide the T cell mediated immunity. Another thing I want to discuss, upon antigen stimulation, naive T cell are activated and express the subset cells via the cell division and express specific surface receptors. So in effector cells, they have the TCR. Without TCR, they will not work. So you can understand there are the three processes are there of the T cell, naive T cells. They will change into the effector cells. So you can understand here is the first type of the process are there, second type of the process are there, and third type of the process are there. It is a naive T cells. Naive T cells are there. They have they will encounter with to the peptides or MSC, and it will undergo to the apoptotic cells, or it is the energetic cells. So these are the different type of the cells are there. It will generate, or ultimately they will not involve into the cell mediated immunity. They will not kill the pathogens. It will go to the cell death through the apoptotic cycles, or you can say there is the energetic cells are there. They lies into the bodies, but they are not differentiated, and they are not activation involves in this, or ultimately maturation will not take place, or it will not able to kill the pathogens. Another process are there. Here is the naive cells are there. They have the peptides and MSC molecules on their dendritic cells. It is a mature dendritic cells are there, and co-stimulation also will take place, or it will cells which are undifferentiated. These are the cells are there. They are undifferentiated. They also involve they are not involved into the cell mediated immunity, or ultimately they will not kill the pathogens. And third type of the process are there. They are able to generate the effector cells. Here is the naive cells are there. It will counter with to the pathogens, or MSC. They have the particular peptides are there, and co-stimulation will take place. And here is the signal 3, like interleukin 12, which are involved, or ultimately it will change into the effector cells. And effector cells, they express the specific surface receptor on the effector T cells, which are involved into the T cell mediated immunity, or ultimately they are able to kill the pathogens. So it is a the naive T cells, they are able to convert into the effector T cells. Another thing I want to discuss, that is known as the T cell receptors. The T cell receptors which are found onto the T cells. You can see it here is the T cell plasma membrane. It is a plasma membranes are there. Here is the transmembrane region, intracellular domains. So these are the made up with the particular proteins. Here is disulfides. There are the two chains are there. One is the alpha chain, second is the beta chains, and constant regions are there. These are the very important region, that is the constant region. Here is the variable regions which are present onto the cell surface. Surface up to the T cells. Here is the antigenic binding sites. It is a variable regions are there. So you can say it is a T cell receptor. It involved into the binding between the antigen presenting cells. And T cell develop a specific receptor for to the recognition of MSC associated foreign antigen peptides and processed by antigen presenting cell. The number of the antigen presenting cells are there, like B cell, like dendritic cells, and macrophages cells are there, they have the particular receptors are there. In particular receptor, MSC are associated with the foreign antigenic peptides, and T cell on the membrane surface have a specific receptor called T cell receptor. So you can say it is a T cell receptor. Without this receptor, it will not activate, or it will not mature, or they are not able to bind with to the MSC associated foreign antigenic peptides, or the reaction will not take place, or ultimately they will not able to kill the pathogens. And TCR consists two polypeptide chains. One polypeptide chain is alpha and second is the beta, and which diverge in nature and bind to the epitope of to the various antigen processing by the APC, antigen presenting cells. So it is a T cell receptors which are present on the T cells. They are involved in the binding with the antigenic peptides which are associated with the MSC molecules. Another thing I want to discuss, the T cell classification, how the T cell are classified. All T cell produce different protein molecule on their surface. A lot of protein molecules which are expressed onto the T cells, and these different surface protein known as to the CD. CD is cluster of differentiation. So it is in different type like CD40, CD28. So basically it is a different surface protein known as the CD. CD, it is a different type of the CDs. We can distinguish the different type of the cells on the basis of to the cluster differentiation number, CD numbers. Each cell surface protein is assigned into the different number, which help to the identification of various type of to the white blood cells. It means the T cell or white blood cells are there. They have the different markers are there. So we can identify on the basis of to the marker which are present in the form of the CD. And T cell are divided into two classes. And first one is the cytotoxic T cells, and second is the helper T cells. Based on the surface protein, it is divided based on to the surface protein CD8 positive and CD4 positive cells. You can say it is the cytotoxic T cells and helper T cells are there. I want to discuss both cell are there, cytotoxic cells and helper T cells. So these are the cytotoxic T cells are there. T cell express a CD positive surface marker. You can see it is a cytotoxic T cells, and they have to the markers like CD8. Another is the T cell receptor bind to the MSC class first. So CD8 positive cells are there. They are able to bind to the MSC first molecules. The T cell express CD8 positive surface marker to recognize the MSC first molecule associated antigen peptide present on the antigen presenting cells, and therefore CD8 positive bind to the MSC class first, ensure the cytotoxic T cells only interact with to the infected target cells. The infected target cell, it may be bacteria or it may be viruses are there. The cytotoxic T cell release, this cytotoxic T cell, they are able to release proteolytic enzyme to destroy and kill the antigens infected cells. It may be viral peptides or bacterial peptides are there, or they will kill it. Hence they are known as the killer T cells or cytotoxic T cell. Another cells are there that is known as the helper cell. According to the name, they are able to help in the regulation of other immune cells such as to the T cell, B cell, dendritic cells and macrophages. Hence it is the called T helper cell. They are able to help to the different type of the cells are there. And T cells, they have the helper T cells, they have the CD4 positive markers are there. Here is the TCR, T cell receptor, bind to the MSC class second. So T cell which express CD4 positive marker and interact with the MSC class second molecule, associated antigen peptide present onto the APCs, present onto the antigen presenting cell. It may be macrophages or it may be dendritic cells, and B cells are there. The number of the APCs are there. They express the MSC class seconds, and CD4 positive cell bind to the MSC class second, ensure T helper cell which interact with to the antigen presenting cell and secrete to the signal molecule like cytokines. So after binding of the TCR with the MSC, they are able to produce the signal molecule that is known as the cytokines. Cytokines, it help in the differentiation, maturation and activation of the T cells. Another thing I want to discuss the difference between the cytotoxic T cells and helper T cells. It is a class cytotoxic T cell and helper T cells, and it is a CD cluster of differentiation molecules. They have the CD8 positive cell and CD4 positive cells in case of the T helper cell. In case of cytotoxic T cells, they activate by the MSC class first molecule onto the APCs or infected nucleated cells, and helper T cells they activate through the MSC class second onto the APC or you can say the antigen presenting cells. And function, it directly kill to the pathogen infected cell, release proteolytic enzymes and kill antigen infected cell, or it may be viral peptides or bacterial peptides are there. The T helper cell, they are able to regulate the humoral and cellular immunity, and activated B cell produce antibodies and cytotoxic and macrophages and NK cells are there. Macrophages and NK cells are there. So it is a difference between the cytotoxic T cells and helper T cells. Another very important things I want to discuss, without the antigen presenting cells, they are not able to distinguish by the T cell, and T cell receptor, they can bind with the antigen presenting cells. Basically in this case, antigen, they are able to present the antigens. Antigen presenting cells, they are able to process your antigens. It may be bacteria or it may be viruses are there. Basically the foreign molecules are there. It will present. So I want to discuss the different characteristic and their features of the antigen presenting cells. The antigen presenting cells, basically three antigen presenting cells are there. One is the dendritic cell, another is the macrophage cell, and third one is the B cell. The B cells, you can see there are the three cells are there. The antigen presenting cells are group of the immunological cells that display antigen to the T cells. So basically these are the dendritic cells are there. It is viruses. The viruses it will engulf by to the dendritic cell, or ultimately they are able to process, or it will present through the MSC molecules. And antigen, basically another is the microphages are there. Microphages, they are able to engulf the bacteria. You can see in this figure the bacteria are there. It will engulf by to the microphages, or ultimately they are able to process it, or it will present on the surface of to the macrophage cells, or it will recognize through the T cells. And third one is the B cells. These are the antibodies are there. Here is the microbial toxin. They are able to bind it. And APC are involved into the intracellular digestion of the foreign antigen such as to the virus, bacteria, etc. The dendritic cell, macrophages and B cell also serve as the APC, antigen presenting cell. So your antigen presenting cells are there, are the three type of the antigen presenting cells are there and present in the high number in the lymphoid organs. And antigen presenting cell are necessary for the initial activation of the naive T cell. The naive T cell, it will activate only with the help of to the antigen presenting cell. If antigen presenting cell was not there, the naive T cell, it will not change into the effector molecules like cytotoxic T cells and T helper cells. Another thing I want to discuss, the T cell and recognition of the APC, antigen presenting cells. You can understand through this figure, it is a APC, antigen presenting cell. Basically dendritic cells are there. Here is the T cells. It is a T cell receptor. They are able to recognize the MSC, either MSC first or MSC seconds are there. So these are the another cluster differentiation molecules are there. They can recognize it or ultimately activation will take place. So it is a T cell and recognition of the antigen presenting cell. T cell only recognize intracellular antigen peptides bound to the MSC molecule. If the antigenic peptides which are not bound onto the MSC molecule, they are not able to recognize it, or ultimately your T cell activation, maturation and differentiation will not take place and T cell will die. An MSC molecule and antigen presenting molecule express on all the nucleated cell. So your MSC molecule, these are the MSC molecule, it will present only the all present onto the all nucleated cells. MSC is the larger collection of the gene encoding. It is found in all nucleated cells, and they are divided into two classes, MSC class first and MSC class second. Both are involved into the T cell mediated immunity. Another thing I want to discuss, MSC peptide complex express onto the APCs interact with to the T cells. And MSC class first molecule are present onto the all nucleated cell. I have already discussed it. Your MSC class first molecule are presented in the all nucleated cells and present normal self and non-self antigens to the effector T cells. On the other hand, MSC class second molecule are only present onto the surface of antigen presenting cells like macrophage, dendritic cells and B cell. And they present abnormal or self non-self antigen peptides for the initial activation of the T cell. The MSC class first interact with only to the CD8 positive cytotoxic cells, and MSC class second molecule bind to the CD4 T helper cells. So these are the mechanisms are there. It is the helper T cells are there. Here is the cytotoxic T cells are there. They are able to recognize the MSC molecule. MSC first molecule are there. Here is the MSC second molecule, or ultimately activation, maturation and differentiation will take place of the T cells. The interaction is very important. Without interactions, the T cell will not work. Another is the antigen processing and presentation via MSC to the T cells. So you can see this the cells. It is a dendritic cells are there. Here is the antigen MSC class first and seconds, immunological epitopes. Here is the lysozymes. Basically the bacterial pathogens are there. They are able to infect to the dendritic cells, and here is the engulf, they are able to engulf it, or ultimately process will take place and they will present onto the MSC molecules. And the step involved into the, there are the three steps which are involved in antigen processing and presentation. First is the when bacterial pathogen interact with to the dendritic cell, it engulf with to the phygosome via the phagocytosis, and the phygosome fuse with to the lysosomes and hydroxylations of the foreign antigens into the smaller peptides. Antigen peptides along with to the MSC molecule are then displayed for to the T cell recognitions. So your peptides, basically antigenic peptides, which they can process through the APCs, or ultimately they will present it, or it will recognize by to the T cells. And APCs primarily ingest and digest non-self antigen by the phagocytosis and process of antigenic peptide with the class second molecules, MSC class second molecule for presentation of the T cell activation. MSC molecule encode a large group of the genes. So your MSC molecule, they are able to code the large group of the genes are there, and they will express it, or ultimately they will recognize it to the T cells. All healthy nucleated cells have MSC class first molecule, and that are essential for presentation of the normal self antigens. When the MSC class second molecule are found only on the surface of to the antigen presenting cells, which play important role in the T cell activations. Dear viewers, I want to summarize the part first. You have learned the various things like mature or naive T cell migrate from red bone marrow to the thymus for the maturations, and antigen presenting cells present MSC peptide complex to the T cell and their activation that lead to the cell mediated immunity. T cell activation required the three main components. First one is the T cells, and second was the MSC complex, and third one is the antigenic peptides. Then your T cell will activate, otherwise your T cell will not activate. And you have also learned the T cell express co-receptor on the surface such as to the CD4 positive and CD8 positive, which facilitate TCR engagement with the peptide MSC complex on APCs. And you have also learned the T helper cells carry co-receptors and CD4 positive that recognize MSC class second molecule, while co-receptor CD8 positive that recognize MSC class first molecule that initiate cell mediated immunity. Dear learner, I want to take a quick break. Don't go away. We will back within 2 minutes. Thank you. Dear learner, welcome you all in the second session. You can say the part second. In this session, I will discuss in detail the activation of the T cells and maturation of the T cells and differentiation of the T cells. And finally, I will discuss in detail and memory cells, how develop the memory cell, what is the role of the memory cell inside to the body. So I want to discuss one by one the activation of the T cells. For activation, T cell must interact to the antigen presenting cell. Without interactions, the T cell will not activate. And mature naive T cell circulate continuously into the secondary lymphoid organs. Secondary lymphoid organ, it may be lymph node or it may be spleen are there. And they will continuously circulate in the lymphoid organs by the blood stream to identify antigen infected cell and return back to the blood cells. And the T cell only recognize antigen peptide which are properly presented, properly processed by the MSC molecules. If MSC molecule they are not able to process it, T cell will not recognize, activation will not take place. The T cell do not attain the antigen without MSC, and so that it is known as the MSC restricted cell. So you can say the T cell, it is a MSC restricted cell. Without MSC, they will not activate. When the mature naive T cell recognize a specific antigen peptide MSC complex on the surface of the antigen presenting cells, and activation process is started. It means you can say your T cell is MSC restricted molecules. It will interact with the APC, then activation will start. Otherwise, the activation of the T cell will not activate. And there are the 3 steps involved in the T cell activations. Step first, step second, and step third. In case of step first, the T cell co-receptors recognize a specific antigen peptide present by MSC molecule on the surface of the APC. So basically APC are there, antigen presenting cells are there, they are able to present the peptides, antigenic peptides through the MSC molecule. So it is a first step. And second step, CD4 T cell receptor bind to the specific antigen or peptide MSC complex, and while CD8 positive cell bind to the peptide MSC class second complex. So these interaction is very important. Without this interaction, the T cell activation will not take place. And step third, upon proper binding and interaction, antigen presenting cell deliver 2 distinct signal. One to the TCR by the peptide MSC complex, and second is the co-stimulatory signals like cytokines that trigger the activation of the naive T cell. Otherwise, your naive T cell will not activate. There are the 2 signals are there. One is the co-stimulatory signals, and another signals that come from the TCR. And TCR, they are able to bind to the peptide MSC complex. So these are the 2 signals which are very important, then activation will start. So you can understand through this figure, these are the T cells. They have the particular receptors are there, that is known as the T cell receptors. Here is the cells are there, and they will recognize it, or ultimately they will activate. So these are the 3 steps which are involved into the activation of the T cell activations. The activate naive T cells secrete signal molecule cytokine that trigger activation of the T cell maturations. So signals, secrete signal molecule, basically cytokines are there. IL-12, which are very, very important, which are involved into the T cell activation or T cell maturation. The activate selected mature T cell undergo proliferation and express their surface receptor molecules, and the surface receptor molecule they will express it, that is known as the CD, and CD40, CD28, and number of the surface molecules are there, surface receptor molecules they are able to express on the surface of the T cells. And some T cells where only CD8 positive marker, which are functionally recognized as a cytotoxic T cells, and some T cell have CD4 positive surface marker on their surface, and such cell are called CD4 positive helper T cells. The key differences only is that the CD4 positive cell recognize the foreign peptides by the MSC second molecule, and while CD8 positive T cell recognize antigenic peptide by the class MSC class second molecules. So it is a differences between the CD4 positive T cells and CD8 positive T cells. And another thing I want to discuss, the maturation of the T cells. And thymus is the main site for to the T cell maturation. So your T cell they are able to mature in the thymus. The thymus is the main site for to the T cell maturation, and mature T cell serve as the progenitors cells or naive cells, as they have not been presented to the antigenic peptide or MSC complexes. Thymus execute the mechanism for to the selection of effective and non-effective T cells. So here is a role of thymus is the very crucial. They can execute it, the mechanism for selection of the effective and non-effective T cell. It provides a suitable microenvironment up to the T cell development. I have already discussed in my previous lecture. It is divided into the positive selection and negative selection, which are collectively called the thymic selections. So your T cell maturations, it will mature it via the positive selection or the negative selections. It is also known as the thymic selection. So I want to discuss what is the positive selection and what is the negative selection, what is the role of the positive selection and negative selection in the maturation of the T cells. So here is the positive selection of the T cells. Occur in the thymic cortical region of the thymus, where the mature T cell interact with to the cortical epithelial cell having MSC. Here is the role of MSC is very important. Your T cell they will interact with the thymic cortic regions of the thymus, or they are able to mature it. Only few T cell are positively selected, and rest of T cell dies. About 98% T cell dies, and through the apoptosis. Apoptosis is the programmed cell death which are involved in this case, or they in this case the T cell will die. Only 2% cell which are involved in the maturation. During to the development, and mature T cell express surface receptor along with to the co-receptor in CD4 positive and CD8 positive, that is known as the double positive selection. So here is the role of the CD4 positive and CD8 positive of the co-receptors are there, that is known as the double positive T cells. This is very important process are there. Your T cell they will mature with the help of this mechanism. Only those mature T cells are positively selected if TC are recognized properly and interact moderately to the self peptide, which are self MSC complex presented by the thymic cortical epithelial cells are there. So it is a very important process. They are involved into the positive selection of the T cells, and this interaction release survival signals that prevent T cell from the undergoing to the cell death, and T cell whose the receptor do not interact with the cortical epithelial cell, and they would undergo to the cell death by apoptosis. In the positive selection, T cell undergo the rearrangement and proliferation by the mitosis, and T cell differentiation into distinct type of the CD8 positive cells, that is known as the cytotoxic T cells, and CD4 positive cell, that is known as the T helper cells, are there, and their differentiation will take place. And these two distinct mature naive T cells can proliferate into the and differentiate into the subtypes when they interact again with to the specific MSC associated antigen peptides. So you can see it here is the T cell precursor are there. They have to the different marker CD, a cluster of differentiation CD8 and CD4 positive, and T cell receptors are there. These are the different CDs are there, and if they will encounter, they will bind to the antigenic peptides, they will mature it, otherwise maturation will not take place. Here is the death by the apoptosis will take place of your T cell, which are which I have already discussed it, how the death take place are there. Another is the negative selections are there. Here is the negative selections. The positive selected cell further undergo the negative selections. Your positive you can understand through this pathway. It is a positive selection. Positive selection undergo the negative selections. The medulla region of the thymus is the committed to the negative selections. In this process, mature T cell are again presented, and the cell peptide MSC molecule complex derived from the thymic medullary epithelial cells. If the TCR, T cell receptor, bind tightly to the self peptides, so here is the, you can understand, here is the T cells are there, T cell receptor are there. If they bind tightly to the cell peptide MSC complexes, then T cell receives suicide response to the cell death by the apoptosis process. Only those T cell will survive which possess the minimal affinity. If affinity is the maximum, they will go to the apoptosis process, and here is the minimal affinity are there to the cell peptide and self MSC molecules. So that is known as the negative selection of the T cell. The negative selection of T cell are autoimmune cells, which can be harmful for to the normal body cells. To overcome this problem, such autoreactive thymocytes are removed by to the negative selection. Here is a very important process are there. By using of this, we can overcome of these problems, and thus thymic selection ensure the selection, maturation, expression of distinct class of to the T cell receptor on their surface. The T cell express different T cell receptor to recognize cell peptides, non-cell peptide bound to the MSC molecule presented by to the antigen presenting cell and nucleated cell. So these are the basically negative selection of the T cell, which are very, very important, or ultimately we can remove these problems, and this process is they are able to select the CD4 positive cell or CD8 positive cells. Another is the differentiation of the T cells. As the T cell become properly mature by the thymic selection. The thymic selection, which are very, very important, by this process the T cell become properly mature, and they migrate to the thymus into secondary lymph node, which they interact with to the antigen presenting cells. So here is role of antigen presenting cell is very, very important. Your T cell, they will interact it, or ultimately differentiation will take place in the thymus. The thymic selection type of the process which are involved. And following activation, T cell can differentiate into the subtypes which are known mature CD4 positive TCR recognized specific antigen peptide MSC second complexes, and CD4 positive T cell receptor recognize specific antigen peptide MSC class first complexes. However, T cell differentiation can take several days or weeks for making their subtype. Another is the T cell helper T cell differentiation. So you can understand by this figure, it is the naive helper T cells are there. It is activated T cells. They are able to bind with the dendritic cells. In dendritic cells, they have the MSC class second molecule. Here is antigen extracted from the pathogens. So here is the peptides are there. It is extracted from to the pathogen, or ultimately they will differentiate into the T helper 1 cell and T helper 2 cell and memory T cells. So they are able to differentiate into the three type of the cells are there: memory T cells and T helper 2 cells and T helper 1 cells. And after being activated, T cells undergo clonal selections by the proliferation. So it is a clonal selection and proliferations are there and differentiated into distinct subtypes. And there are the three type of the cells are there: memory T helper cells and T helper 2 cells and T helper 1 cells are. All these T helper cell subtypes are characterized by to the expression of the unique cell surface. It is a particular cells like T helper 2 cells, they have the specific receptors are there. So on the basis of the particular receptor, we can recognize it, the memory cells and T helper first cell and T helper second cells. In the proliferation and differentiation of the distinct T cell lineage depend on to the complex of the cytokine signaling. And another thing is which are very important, the cytokines, they are involved in the proliferation and differentiation. It will release by to the particular cells that is known as the antigen presenting cells, and they will secrete the cytokines, and cytokine signaling which are involved with, or ultimately proliferation and differentiation will take place. They are involved into the activation, proliferation and differentiation of the T cells, CD4 positive T cell and CD8 positive T cells. And interleukin 12, it is very important cytokines are there which are released by to the dendritic cells. They are able to stimulate and activate T helper cells to produce T helper subset and T helper 1, T helper 2 and memory cells and regulatory T helper cells are there. In interleukin 10 also help to the develop up to the T helper 2 cells. And another is cytotoxic T cell differentiation. It is another type of the cells are there, cytotoxic T cells. You can say it is a killer T cells are there. Like to the T helper cell differentiation process, peptide MSC complexes and co-stimulatory cytokine initiate the cytotoxic T cell differentiation process. So here is the MSC class first complexes are there. They will recognize by to the T cell receptors. Here is the T cell receptors are there. They have to the CD8 positive cell MSC molecules. They will recognize it, or ultimately the T cell differentiation will take place. Activated T cell act as a effector cytotoxic cells to kill the antigen infected antigen infected APCs, and T helper 1 cell secrete cytokines and stimulate the proliferation and differentiation of to the cytotoxic T cell. And effector T cell, you can say it is a activated T cells are there, or you can say it is a effector cells. Effector T cell release cytokines molecule called the called perforin and granozymes that destroy to the antigen infected cell. So it is a antigen infected cells are there. They will recognize through the effector T cells, or they will release perforin and granozymes, or ultimately the antigen infected cell will die. Effector T cell differ from to the naive mature T cell. They respond quickly and attack to the target cell and destroy to the intracellular antigen without further differentiations. And these effector CD8 positive cell become memory cells and ready to the respond to the subsequent exposure up to the specific pathogen. Differentiation step is required to make clonal T cell and a specific function up to the regulate to the prevent microbial infections as well as development of the memory cells. Another thing I want to discuss, you have seen related to the T cell activation, T cell differentiation and T cell maturation. It is another very important cells are there, that is known as the memory T cell. Here is the naive T cells, they are able to produce the memory T cell progenitors, and it is effector cells are there. Effector cells, they are able to kill the pathogens, or it ultimately the cell death will take place. Here is the memory cells, they are able to generate memory cells. Another is the pathogen encounter and the cell division will take place. They will divided into two group. One is the cells, the death take place. Here is the memory cells are there. One effector T cell are fully developed over to the period of time. They produce memory cell by a clonal selection or cell division. Memory cell shows the immune response when to the antigen infected cell second time, and they have memory cell for recognizing and kill to the specific antigen infected cells, and the memory cells are considered as a secondary immune response. And memory cells are long-lived immunological cell and produce quick immune response upon re-exposure to the antigens. Memory cells are memory T cell are a specific one of the particular type of antigen. Memory T cell were either CD4 or CD8 marker but not both. So your memory cells, they have only CD4 positive or CD8 positive markers are there, but not both. These mature cells further proliferate into effective CD8 positive and cytotoxic T cells and T helper cell that target and kill the pathogens. Dear learner, I want to summarize part second. You have learned the positive selection ensure the survival of only those T cells whose the TCR recognize self-MHC molecules. Otherwise, they will not survive it. The T cell will die it. So it is a very important process in the T cell activation, T cell differentiation and T cell maturation. The negative selection eliminate those cells whose the TCR react strongly with the self-peptide MHC molecule. Thereby they inhibit self-reactive immune response or autoimmune cells. T cells are there. Another, you have also learned the activated helper T cells can differentiate into their various subset such as to the T helper 1 and T helper 2 are memory cell. So basically T helper 2 cells, they are able to produce the cytokines, or they can regulate the T cell activations. Memory T cell are a specific program immune cells that responds quickly to the secondary antigen exposure. Cytokines are proteins that act as a signaling molecule for that play important role in the activation, differentiation, and regulation. Dear learner, if you want to clarify each and everything, you can go through the IGNOU study learning materials BZYET 141. So it is the unit number 11s. You can go through, you can clarify each and everything. If you feel any problems, you can mail me at cram1@gmail.com. Thank you.